Well, this is a very timely topic for me because we're on the verge of introducing arrays for clinical testing in the cytogenetics lab at Children's Hospital. And this is a time of great change for the field of cytogenetics with the introduction of arrays. And it's a very exciting time to be a cytogeneticist right now. The learning objectives for this presentation are to understand array-based comparative genomic hybridization technology. And Array Cgh is the array-based technique that's being used in cytogenetics. You should be able to list the advantages and limitations of Array Cgh. I'll describe some of the recent discoveries that have been made using this technology. And then I'll also talk about applications of Array Cgh in the clinical lab. So before I jump right into the arrays, though, I wanted to give a brief history of cytogenetics because it's interesting to see how things have evolved to where they are today. So way back when people first started looking at human chromosomes, the preparations that they had were so limited that they had trouble even counting the number of chromosomes in a nucleus. So this led in 1923 to a publication that actually stated that the human chromosome number was 48. And this could not be unequivocally challenged for a number of decades. And things really didn't improve until the early 1950s when a cytogeneticist by the name of T.C. Hsu made a very significant discovery that revolutionized the field of cytogenetics. And what's interesting about this discovery, it was actually the result of a laboratory error. So what happened was one of the techs in the lab made up a solution incorrectly. And the solution was very hypotonic with respect to the cells. And when the cells were incubated in it, it caused them to swell. And they got much better chromosome preparations this way than they had ever seen before. And this lab error then led to a number of very significant discoveries, including the correct human chromosome number, which we know now is 46. And once it was determined that the human chromosome number was 46, then it was found that abnormal numbers of chromosomes could give rise to human disorders. And the first of these findings was that Down syndrome was due to an extra copy of chromosome 21. And then in 1960, Knoll and Hungerford discovered the Philadelphia chromosome as a recurring abnormality in chronic myelogenous leukemia. And this was prior to the time that chromosome banding was introduced. So they actually initially thought that this Philadelphia chromosome could be a deleted chromosome 21. And it wasn't until a number of years later when chromosome banding techniques were introduced and then cytogeneticists could begin to see more subtle abnormalities in the chromosomes. So a few years later, Janet Rowley figured out that the Philadelphia chromosome was not a deleted 21, but that it was actually a balanced reciprocal translocation between chromosomes 9 and 22. And in the early to mid-70s, it was determined that amniocentesis was a safe and accurate method for prenatal diagnosis. And this allowed cytogenetics to be brought into the clinical lab as a diagnostic tool. And then by making some modifications to procedures, more high-resolution G-banded karyotyping was achieved. And through tweaking over the years, we've finally gotten to the point where we can see changes on the G-banded chromosome that are at the level of about 3 to 5 megabases. So in other words, in order for you to see a deletion on a G-banded chromosome, it has to be at least about 3 megabases in size. So that's still pretty big. And you have to also keep in mind that this is in our very best preparations, which are stimulated peripheral blood lymphocytes. When you're looking at things like amnios and bone marrows and solid tumors, the resolution can actually be much worse than this. So we knew we were missing a lot of things just by looking at G-banded karyotypes. But luckily for cytogenetics and for patients, in situ hybridization was developed. And this allows us to detect abnormalities at a much better level of resolution than we can with just standard cytogenetics. So by FISH, you can actually detect a deletion that's on the order of 50 or 100 kilobases. And initially, in situ hybridization was developed using radioactive probes. And this isn't very practical for use in the clinical lab. not only because it involves the use of radioactivity, but also the exposure times were quite long, and there was a lot of background. And you couldn't localize the signals very well on the chromosomes. But then probes could be fluorescently labeled, and initially they were indirectly labeled, usually with biotin or digoxygenin, and then they were then detected with a secondary antibody with lots of washes in between. And then finally, direct labeled probes were introduced, and this really streamlined the process and made it valuable for the clinical cytogenetics lab, so that over the years, the number of FISH tests has increased dramatically. And like G-banded karyotyping, it's an indispensable part of what we do now. So this is an example of one of the more common constitutional abnormalities that we see in the clinical lab. This is the 22q11.2 deletion that gives rise to a variable phenotype. Sometimes it's recognized as velocardiofacial syndrome. Sometimes it gives rise to DeGeorge syndrome. And shown here are 3 pairs of chromosome 22, and the right chromosome in each pair contains a deletion, and the arrow points to the region of deletion. And I think you can see by this that this is a very subtle abnormality, and it's difficult to detect on G-banded chromosomes. And this deletion is typically 3 megabases in size, so it's right at that limit of resolution of what we can get with G-banded cytogenetics. And it's been estimated that we can only see this on the G-banded chromosomes about 30% of the time, that it's actually present. So now the standard of care is to diagnose this deletion using FISH. So this is the normal 22 here with the orange probe in the critical region for the deletion, and the green probe is a control probe. And this is the deleted chromosome 22 here. And you can see the absence of the orange signal that indicates the deletion. And so now we have dozens of probes in the clinical lab that cover not only all the microdeletion and duplication syndromes, but also the subtelomeric regions, because we know that rearrangements in the subtelomeres often give rise to mental retardation. And we also have probes that cover most of the rearrangements that are seen in hematolymphoid disorders. The one problem with FISH is that it's a very targeted study. So we're only looking at one or a few targets at a time by FISH. So you really have to know what you're looking for in order for FISH to be useful. So the clinicians who are looking at a kid with an abnormal phenotype have to be able to recognize what phenotype they think that child has and tell us what probe to use. And what's so great about the arrays now is that it's like doing hundreds or thousands of FISH tests all at once. And that's why we're so excited about them. In addition to FISH, there are also more advanced molecular cytogenetics techniques. One of these is spectral karyotyping, that's also known as SCI or MFISH. And this is a 24-color FISH technique that allows you to visualize all of the chromosomes in different colors with a single hybridization experiment. And this technique can be very useful for things like characterizing translocations when you're not sure what chromosomes may be involved. And it's also useful for characterizing marker chromosomes. Markers are pieces of chromosomes that we can't identify by G-banded analysis. But practically speaking, this technique really never gained widespread use in the clinical lab. The SCI instrument is very expensive. The probes are expensive, and it's somewhat of a cumbersome, lengthy technique to perform. And there's really only a limited number of scenarios that we would use it in. So we have a SCI system over at Children's, and we probably only use it a handful of times a year now, primarily to try to characterize marker chromosomes that we see. And there's other ways that you can do that. So there aren't a lot of clinical labs that are using this to any great extent now. Another advanced molecular cytogenetic technique is comparative genomic hybridization. And when this was initially introduced, it was a chromosome-based technique. But then in the late 1990s, Dan Pinkel and his colleagues adapted this to arrays. So this is what chromosome-based CGH looks like. With this, you have a test DNA, and this usually consists of, it can either be a tumor or it could be DNA from a patient who has mental retardation and congenital anomalies. And you have a reference DNA, which is typically a normal DNA sample. And you label your test DNA in one color and your reference DNA in another color. Typically, the dyes that are used are Cy3 and Cy5. And you mix these DNAs together in equal amounts, along with COT1, which blocks repetitive DNA sequences. And you hybridize this mixture to normal chromosomes. So you don't need chromosomes from your case of interest. You just need a source of normal chromosomes. And then anywhere in the genome where you have an equal amount of test and reference DNA, you'll see a yellow signal. Anywhere where you see an excess of red signal, it indicates that you have a gain in your test DNA compared to your reference. In this case, if your test is in red and your reference is in green. And anywhere in the genome where you have an excess of green hybridization signal, it means you have a loss of chromosomal material, or DNA, in your test compared to your reference. So this is what an actual chromosome CGH experiment looks like, and this experiment was conducted by Kent Opheim a number of years ago. Kent is the other director of the Cytogenetics lab over at Children's Hospital, and this is what an example of what a normal male versus a normal female hybridization looks like. So you can see that the majority of the chromosomes give a yellow hybridization signal indicating that there's equal amounts of test and reference DNA. But you can see the difference in the X and the Y chromosome indicating the difference in 2 X's in female versus XY in male. So chromosome-based CGH is a very good genome-wide screen for chromosome gains and losses. And the big advantage of it is that you can use formalin-fixed paraffin-embedded tissue. So again, you don't need metaphase spreads or chromosomes from your case of interest. In order to get chromosomes or metaphase spreads, you have to have living cells that need to be cultured. So a big advantage of this technique is that you can retrospectively go back and look at archival material. So this was a very useful technique for studying solid tumors. One slight disadvantage of it is that you do need high-quality normal metaphase spreads. And this isn't a problem for clinical labs, but a lot of research labs find it difficult to do this. So it wasn't widely used in a lot of different labs. And the biggest limitation to it is that it doesn't have very good resolution. So if you're looking for single copy gains and losses, you have to have a change of at least about 10 megabases in order to detect it. So that's a very big piece of DNA. Sometimes you can see smaller regions of gain if there's a high-level amplification in a tumor event. But typically, if you're looking for single copy changes, the resolution is very poor. And the other big disadvantage of it is that it can't detect balanced chromosome rearrangements. So again, this was not a technique that ever gained widespread use in the clinical lab, primarily because it has these limits of resolution and it can't detect balanced rearrangements. However, CGH is now performed on arrays, and this technique looks similar to chromosome-based CGH in that you have a test labeled in one color and a reference DNA labeled in another color. You again mix these together in equal amounts with a blocking agent, but now instead of hybridizing to chromosomes, we hybridize to arrays. And the arrays have hundreds or thousands of DNA sequences spotted on them. And again, anywhere where you get equal amounts of test and reference DNA, you end up with a yellow hybridization signal. Anywhere where you have an excess of green, in this example, it indicates that there's a loss of chromosomal material in your test compared to your reference. And again, anywhere where you have an excess of red signal, it indicates that you have a gain of chromosome material in your test compared to your reference. So this is what an actual Array Cgh experiment looks like, and you might say this looks the same as an expression array. And it does. However, it's very important to keep in mind that these are two extremely different techniques. So Array Cgh is a DNA-based technique, whereas expression arrays utilize RNA. So we don't have a lot of the same problems with Array Cgh that you can encounter with expression arrays. DNA is much more stable than RNA is, so we don't have to worry about degradation as much. And we don't see fluctuations in DNA copy numbers like you can see fluctuations in gene expression levels that are due to things that are difficult to control, like ischemic time of tissue and hundreds of other variables. The other thing that's important to keep in mind is that Array Cgh detects DNA copy number changes. So it can detect gains in the genome, in other words, duplications or amplifications in tumors, or gains of whole chromosomes like trisomies. And it can also detect losses, so deletions and monosomies. Whereas with expression arrays, you're looking for changes in gene expression levels. And sometimes the two of these will correlate. So if you have a region of gain in the genome, it's not uncommon that many of the genes in that region will show increased expression. Similarly, if you have a loss of a chromosome region, it's not uncommon for genes in that region to show decreased expression, but there's not always a perfect correlation between those two. And compared to G-banded karyotyping, there are a number of advantages of Array Cgh. Again, it's a DNA-based technique, so you can use formalin-fixed paraffin-embedded tissue if you have to. You can also use very small starting amounts of DNA because it works very well with whole genome amplified samples. Compared to standard karyotyping, Array Cgh is much more high throughput, and it's also more amenable to automation in the future. But the biggest advantage of Array Cgh over conventional karyotyping is the resolution. So with Array Cgh, your resolution is going to be limited only by the size of the DNA sequences that are spotted on your array and the distance between those sequences. So depending on the type of array that you're using, you can get down to a level of resolution for detecting gains and losses on the order of 50 kilobases, or even less, depending, or even better resolution depending on the type of array that you're using. There are some limitations to this technique, though. Currently, Array Cgh can't detect balanced rearrangements, which is a big disadvantage for us, although Harvey Greisman is going to fix that for us very soon. And this technique also cannot detect tetraploidy. Right now, it's pretty expensive to perform. The chips are very expensive. Hopefully, that will decrease in the future. And one of the big problems right now is that the choice of chips for clinical use is very limited. So for research purposes, there are new chips coming out every day to the point where it's really difficult to decide what chip you should be using. But for use in the clinical lab, there's very few chips available to us. Different platforms can be used for Array Cgh. So initially, when this technique was introduced, people used the same cDNAs that were used for expression arrays. The problem with those is that they don't give very good signal to noise. So, and since there are other arrays that work better now, the cDNAs are pretty much obsolete for use with Array Cgh. Many people are still using BAC arrays. A BAC is a bacterial artificial chromosome, and it's very similar to a plasmid, but it can hold much larger pieces of DNA. So BACs can hold inserts that are on the order of 200 to 300 kilobases. The disadvantage to these is that you have to extract DNA from the clones before you can spot them on the chips. And BACs are low copy number in bacteria, so you don't get very good DNA yield when you do an extraction. And so typically, in order to get enough DNA to spot a reasonable number of chips, you have to amplify the DNA by PCR. So when you make these chips, you run into all the same problems that you do with any other PCR-based technique, and that you can get cross-contamination. Oligonucleotide arrays have been used more recently for Array Cgh, and these work very well. And the oligos can range anywhere from 25 to 60 nucleotides in length, depending on which manufacturer is making them. And the oligos are printed directly on the arrays. So you don't have the same production problems that you do with the back arrays. You don't have to worry about cross-contaminating clones, cross-contaminating PCR products. So I think in the future, this is probably going to be the platform of choice for Array Cgh. And there are some different applications of Array Cgh. This technique is frequently used to identify constitutional microdeletion and duplication syndromes. It can also be used to identify regions of the genome that contain oncogenes or tumor suppressor genes. So regions of gain or amplification in tumors frequently contain an oncogene or more than one oncogenes. And regions of deletion often indicate that there are tumor suppressors there. And recently, it's also been used to study large-scale genomic polymorphisms. So I think most people are familiar with SNPs, or single nucleotide polymorphisms. But using the arrays, it's been found that there are polymorphisms that are present in the genome on a much larger scale. And I'll talk more about that in just a moment. So I showed you what a 22q11.2 deletion looks like on the G-banded chromosomes, and also by FISH. And this is an example of what it looks like by Array Cgh. And the typical way to display Array Cgh data is with a log ratio of the test to reference fluorescence intensity signal that's been normalized. So here, each spot represents a different clone on the chip. And it's also typical to do a dye swap experiment. So you do two separate hybridizations. In one hybridization, you label your test DNA in Cy3 and your reference DNA in Cy5. And then you do another hybridization where you've swapped the labels. And so each plot here, the red plot represents one hybridization experiment, and the blue plot represents the dye swap experiment. And the tip of the short arm is located at this end, and the tip of the long arm is located over here. And the cutoffs for gains and losses that we're using in the lab at Children's are shown by these turquoise lines. And you can see here that there's at least 4 clones in the proximal long arm of chromosome 22 that show this mirror image deviation. And this indicates that there's a deletion here. Here's another example of how Array Cgh can be useful in the clinical lab. So this was a case that was sent in, and this child had mental retardation and multiple congenital anomalies. And all of the chromosomes looked normal except for this one chromosome 6 here. And it was noticed that there was a problem on the end of the short arm, and it looks like there's extra material that doesn't belong here. But based on G-banded analysis, we can't tell for sure what that material actually is. So we end up writing the karyotype like this: 46XY add 6P25. And this is basically cytogenetics shorthand for there's extra stuff at the end of chromosome 6 that doesn't belong there, and we don't know what it is. And it's not very satisfying to write these kinds of karyotypes because they represent an incomplete characterization of the abnormality that's shown here. But if you do Array Cgh on this case, you can immediately see what's going on. So this is the chromosome plot for chromosome 6, and chromosome 4 is down here. So you can see that there's a mirror image deviation on the distal short arm of chromosome 6 that indicates that there's a deletion there. And we already knew that based on the G-banding, based on the abnormality that we saw by G-banding. But it also shows that there's a gain of material on the distal short arm of chromosome 4. So it's very easy to characterize these in one experiment by Array Cgh. And so now we can write the karyotype like this, which is probably equally as confusing for someone who's not a cytogeneticist, but makes us feel a lot better. And the other thing is that as we get better at characterizing these abnormalities, hopefully we'll get better at predicting the phenotypic consequences of these types of imbalances in the future. And if you have really good coverage on your array, you can characterize the breakpoints in these cases with very great precision.
Before you go on, would you be able to explain why it's important that you swap the colors and do it again?
So I think you can see, for example, right here, This is one hybridization experiment. If you only had that, there are a couple clones here that are falling right at our threshold for a gain or for a loss. And so, but when you do the dye swap experiment, which is this blue line here, you can see that these clones have a normal log ratio on the dye swap. So we know that we don't have to worry about those clones being abnormal. So it gives you more confidence.
What's the reason for the difference in results?
Oh, it's just the system is somewhat noisy. So it's not perfect. If everything were perfect, your log ratio for all your clones would fall right on this black line right here. But there's always a little bit of noise. So there have been a number of very interesting discoveries made recently using this technique. For one thing, the etiology of charge syndrome was discovered just recently. And this disorder is characterized by ocular coloboma, heart defects, choanal atresia, retarded growth and development, and genital and ear abnormalities. And Bonnie Pagan had one of the initial characterizations of this disorder back in 1981 here at the University of Washington. However, the cause of this syndrome was not discovered until just recently. So what this group did was they screened a bunch of patients with charge syndrome using Array Cgh. And they identified a deletion in the long arm of chromosome 8 in a few of their patients. And actually, most of their patients didn't have a deletion in that region that you could detect by Array Cgh. But when they sequenced the coding region of all the genes that were in the common region of overlap of deletion in the two patients that had it, they found that a very high percentage of patients had mutations in the CHD7 gene. So this allowed the determination of the etiology of charge syndrome in the majority of patients. Another very recent microdeletion syndrome that's been discovered is this deletion in the long arm of chromosome 17. And this was identified by Andy Sharp, who's a postdoc in Evan Eichler's lab here at the University of Washington in Genome Sciences. And they were using a custom array that was designed in Evan's lab. And they screened a bunch of patients with mental retardation on their arrays, and they found this 17q21 deletion in a number of patients. And not only did they have mental retardation, but they were then found to have consistent phenotypic features. So they were all light-haired, had blue eyes, and had a very bulbous nose and other common features. And this deletion seems to be mediated by what are called flanking segmental duplications. These have also been termed low-copy repeats. And these are stretches of DNA that are at least 10 kilobases in size, and they have at least 95% sequence identity to each other. And down here, there are segmental duplications that are shown in yellow. And what happens is that normally during meiosis, this segmental duplication should align with this one, and this one here should align with this one. But sometimes, because they have such high sequence identity, they can misalign with each other during meiosis. So here you get this one lining up with this block. And then if you have a crossover event that occurs here during meiosis, if you follow the crossover, you wind up with one chromosome that's duplicated, shown here, and your other homologous chromosome has a deletion. And many of the known microdeletion and duplication syndromes that have already been characterized have been found to have these flanking segmental duplications. So in addition to causing mental retardation and multiple congenital anomaly syndromes, these segmental duplications also give rise to copy number changes in other areas of the genome that seem to be phenotypically benign. So if you screen normal individuals by Array Cgh, you will find copy number changes in everyone. And yet some of these don't seem to have any consequence to them. And while some of them seem to be benign, others may be associated with disease susceptibility. And an example of this is that copy number variants in the FCGR3 gene have been shown to predispose to glomerulonephritis in both humans and rats. And these copy number variants can consist of either deletions, duplications, insertions, or they can have very complex structures so that you can see multiple sites on one chromosome or multiple sites on different chromosomes. And these structures range in size from kilobases to many megabases in size, and they frequently contain genes. A recent study that was published in Nature screened a number of the HapMap samples with a very high-density array, and they identified 1,500 different copy number variable regions that they estimated to involve at least 12% of the genome. So these are not rare structures that are present. They seem to be very common. And also just recently, 21 different inbred mouse strains were analyzed using a high-density array, and it was found that there are 80 copy number variants between the 21 different inbred mouse strains that are located on all autosomes. So these copy number variants, they seem to be yet another source of genetic variation in addition to things like SNPs that can contribute to disease susceptibility and that make each one of us unique. So now I'm going to talk about Array Cgh and how it's being used in clinical cytogenetics. And there's quite a debate going on right now about whether we should be using targeted arrays in the clinical lab or more broad genome coverage arrays. So targeted arrays have limited coverage in regions that have known clinical significance. So they cover all of the known microdeletion and duplication critical regions and the subtelomeric regions as well. Whereas the broad genome coverage arrays don't have biased coverage like that. Either you can use a 1 megabase resolution array where you have at least one clone every megabase in the genome. And in order to get that kind of coverage, you need about 3,500 different back clones. Or you can also make a tiling array in which there are overlapping back clones across the whole genome so that there are no gaps in coverage. And in order to get that kind of resolution, you need at least 30,000 backs. But you can imagine the more backs that you add to your chip, the more costly it is to produce and the more issues you have with quality control. So right now, Array Cgh is primarily used to look for constitutional chromosome abnormalities. And a big reason for that is that we can't pick up balanced rearrangements right now in the arrays. And in the clinical lab, other than the constitutional disorders, a big part of our volume are bone marrows. And in those, we're often looking for balanced rearrangements. So currently, it's just not a very useful technique for studying those. And in this country, the labs that are performing Array Cgh clinically are primarily using targeted arrays. And there aren't that many labs that are doing this technique in the clinical lab right now. There's probably at least a dozen, but maybe not more than 24 labs that are doing this right now. Whereas in Europe, they seem to be more comfortable using whole genome tiling arrays. And in this recent issue of the Journal of Molecular Diagnostics, there were two competing articles, one that argued for the use of targeted arrays and another one that argued for the use of whole genome arrays. And the group arguing for the use of whole genome arrays says that, not unexpectedly, they have a higher rate of abnormality detection on their arrays. So if you look at the first 100 mental retardation patients that they screened, they picked up 10 pathogenic copy number alterations, and they estimated that they would have missed 6 to 8 of these abnormalities if they were using the targeted arrays that we're using in this country here. However, on the flip side, Bajani and Schaefer, in their article that argues for the use of targeted arrays right now, point out that there are quality control issues with these very broad genome coverage arrays. So again, if you use a whole genome tiling array that's a back array, you need tens of thousands of clones. And in the database, occasionally some of the clones don't map to where they're supposed to, or some of them will cross-hybridize to more than one region of the genome, so there can be identity issues with the back. And again, there can be cross-contamination issues with the more clones you have, either between the backs themselves or between PCR products. It's obviously more costly to produce that kind of array, and copy number variants can present a big problem. So if you're using a targeted array and you find a copy number change, you can be pretty confident that it's going to be clinically significant because your array is covering clinically significant regions. However, if you're using a whole genome coverage array and you find a copy number alteration, which you will, if it falls outside of one of the critical regions for a currently known microdeletion or duplication syndrome, you're not going to know if it's really responsible for that patient's disorder or if it's just a benign copy number variant. And in the clinical lab, the way that we work that out is that we get DNA from both parents and either run them on the array or do FISH with the clones that are shown to be abnormal. And if one of the parents has the same copy number change that you're questioning in the proband, and the parent is phenotypically normal, then we say it's probably a benign copy number variant. However, if the change is de novo in the proband, then we say it's probably responsible for that person's abnormality. The problem is we can't always get both parents to test in the clinical lab, and it's kind of a lot of work for the clinicians and for the lab too. So then if you can't get both parents, you're left with a copy number change of undetermined clinical significance. And that just creates problems in terms of counseling, and it makes everyone a little bit uncomfortable. And Bajani and Schaefer, they point out in their article that they don't really think that we should never be using whole genome arrays. They think that the timing just isn't right at this point because there's just too much that we don't know about these copy number variants. So they argue that we should just be starting out with targeted arrays and then working our way up to higher density arrays in the future. Other issues associated with the Array Cgh testing is that you have to decide what kind of reference DNA you want to use. So some laboratories like to perform sex-mismatched hybridizations, whereas other ones perform sex-matched hybridizations. The advantage of doing sex-mismatched hybridizations is that it gives you a good internal control, because if you hybridize a male to a female, you know exactly how much your X chromosome should deviate. The disadvantage of doing that is that it can make it more difficult to detect X chromosome abnormalities. And then some labs like to use pooled reference DNAs. So you'll take DNA from 5 normal individuals and mix it together. Some labs like to use single individual reference DNAs. And the argument for using pooled DNAs is that you can dilute out copy number changes in your reference DNA. So you are less likely to pick up copy number variants. The labs using single individual reference DNAs, they say that you get very used to that person's copy number changes and how their DNA behaves on the array. And it's really difficult right now to decide what kind of array platform to use, whether you should be using a back array, an oligo array, what kind of density, what kind of coverage you should have on your array. And it's a moving target right now, and we know that whatever array that we're using today may be completely different than the array that we're using a year or two from now. And for us, the choice of which array to use really came down to regulatory issues. So at Children's, we're using a back array. It's a constitutional array that is very targeted. It covers the microdeletion and duplication regions and the subtelomeres. And most of the labs in this country that are doing Array Cgh clinically are using this chip. And another potential problem with the arrays is that, for example, on the array we're using, there are regions that have coverage that are known to cause adult onset disorders. So for instance, there are clones in the Charcot-Marie tooth locus. And the problem with that is that you're not supposed to test asymptomatic children for adult onset disorders, and yet you may run into the issue that this could be you can incidentally find an abnormality in one of these regions. and then it's unclear whether it should be reported, and it kind of creates a counseling dilemma for your clinicians. And then the other issue that the clinicians are going to have to deal with is whether they want to do Array Cgh on a patient now using a targeted array, or whether they want to wait until there's a higher density array in the future. So if a patient has already had a good G-banded karyotype done and a subtelomeric fish screen and maybe a VCSF fish, it probably doesn't make sense to do a targeted array on them now because they're not going to get much more. So for those patients, it's probably better to wait for a higher density array. But patients who haven't had karyotypes yet, then they're going to have to decide whether they should do it now or wait until there's something higher density available in the future. And a really difficult issue right now is how the FDA is going to regulate the use of arrays in the clinical lab. And no one's really sure. And I'm not an expert on FDA guidelines, but I'm going to talk about what's available for clinical use. So right now, there are no chips for Array Cgh that are FDA approved. And most of the FISH probes that we use in the clinical lab are designated analyte-specific reagents. And so it's not a problem for us to use these clinically. We just have to adequately validate them. Although the ASR rule may be changing in the near future. So it's even tough to know what's going to happen with a lot of the FISH probes. And then there are a few labs out there that are printing their own chips, and so they can run their assay as a home brew test, and so they're pretty much exempt from a lot of the regulations. And then for the rest of us, that leaves us with chips that are either labeled research use only or investigational use only. And at least my understanding of research use only is that we're not supposed to use those for clinical testing. So that basically leaves us with investigational use only, and right now there's only one chip that's labeled investigational use only, and that's the Spectral Genomics constitutional chip. And Spectral Genomics was just recently purchased by PerkinElmer. And with investigational use, we're not told that we can't use it for clinical testing, but there are certain restrictions that are placed on the use of the chip. So in order to use the spectral genomics array right now, you have to sign an investigational use only certificate, and you have to state that you're only using the chip for postnatal testing. And that's not a problem for us at Children's because we don't do prenatal testing. But it is an issue for other labs. And in addition to that, we have to agree to confirm all abnormalities with a gold standard test, which would either be FISH or a G-banded karyotype. And that's not a problem for us because right now we're still requiring a G-banded karyotype to be done with every array. And this is just good lab practice anyway when you're introducing a new technology. In addition to that, we also had to agree to send the company data back on every chip that we run. And that's not really a problem. It's just extra work for us to have to do that for every single patient. And of course, we're doing that on an anonymized basis. And PerkinElmer is using that data for quality control and so that they can monitor the behavior of the chips and replace any clones that aren't behaving the way that they're supposed to. So what happens right now usually is that, and I'm talking about constitutional chromosome studies, patients will usually have G-banded karyotypes, and they may have one or two targeted FISH studies, and then if they're normal, they may have an Array Cgh test ordered. But there are some people who predict that this testing algorithm is going to be flipped in the future. So it may be that in the not too distant future, we'll be doing Array Cgh first on every patient. And then if that's normal, we'll do a G-banded karyotype either to pick up a balanced rearrangement, and we know that de novo balanced rearrangements can sometimes cause problems in people, or to pick up anything else that's been missed on the array. So it's an interesting time for cytogenetics. Right now, I wouldn't want to have to predict what kind of chip we're going to be using a year or two from now, how many arrays we're going to be running in the future, what our reimbursement for this testing is going to be. And so there are a lot of interesting things coming up in the future. And there are other people at Children's who've been very helpful in setting up the array testing. Kent Opheim and Chris Clark and John Schoaf did the validation of the arrays that we're using. And we're currently doing a split sample exchange with Art Brothman's lab at the University of Utah. And they've been very helpful in giving us advice because they've been using this chip for a couple years. So that's it.
We have time for a few questions.
The CGH study be quantified in any way by running a standard curve beforehand? Or I don't know, do you do this beforehand by running reference DNA labeled in both colors and specific ratios, 1 to 10, 10 to 1, that sort of thing, trying to get an idea?
Yeah, so it actually is quantitative. We don't do a standard curve, but based on your log2 ratio, you can estimate what kind of copy number change you have. And one of the things that's kind of a difficult issue, not for constitutional cases so much, but more for tumor cases, is how you determine thresholds for gains and losses. In constitutional cases, it's not that, in non-mosaic constitutional cases, it's not that difficult because if you have 3 copies versus 2, you know what that log2 ratio should be. If you have 1 copy versus 2, that's predictable as well. If you add mosaicism in there, then you start to dilute out those ratios. So you actually have to, you can't just go by the set cutoffs that the software gives you. You have to look at every chromosome plot to see if there may be a mosaic abnormality. And I actually have, this is an example of an Array Cgh experiment done on a colon adenocarcinoma. And this is instead of being a chromosome plot, this is a whole genome plot, so that chromosome 1 is shown here, 2, 3, and X and Y down here. And you can see that there's just a lot of up and down in these cases when they have genomic instability. And it makes it really difficult to figure out what to call a gain and a loss, because the red line represents 2 standard deviations from the mean, and the green line represents 3 standard deviations here. And there's a lot of obvious abnormalities, like this gain on chromosome 2 that wouldn't make either of those cutoffs. So for things like solid tumors like this, people use more complicated algorithms that utilize hidden Markov models or things like that. So it is a quantitative technique.
Aaron,
Yeah,
one of the problems with solid tumors, I assume, is dealing with the tissue heterogeneity. What approaches do you think you'll be using to try to account for the normal cells that are in a piece of tumor tissue to decrease that noise signal ratio?
Yeah. So the question is how do you deal with heterogeneity in solid tumors, not only different clones within a tumor, but also contaminating normal cells that can dilute out your abnormalities. And it's been shown that you can do laser capture microdissection on cases plus or minus whole genome amplification, depending on how much DNA you get, and you can successfully do Array Cgh on those. So that's one way to get around that. Pretty labor intensive. I suppose, like in a hematopoietic disorder, you could flow sort. Yeah.
Okay, so I think what I see as part of the problem with this technique is that the technique is the science, or the technique is ahead of the clinical aspect. And so when you get a fingerprint in the lab, how do you assign clinical significance to that? Or how do you counsel patients? Because we don't even know all that's involved in sort of what the normal spread is for all of us without. So how do you assign?
Right. That's right. So the issue is when we find a copy number change, how do we know that it's clinically significant? And again, this is one of the arguments for not using a whole genome array. So that's why most of the labs in this country are still using very targeted arrays, because we know if the only regions we have on the chip are in critical regions for things like velocardiofacial syndrome, Smith-Magenis syndrome, Wolff-Hirschhorn, we know that if we see copy number alterations in those regions, that those are associated with clinical phenotypes. So, and there are, on these chips, there are a few clones in regions outside of known clinically significant critical regions. And one of the ways that you can try to determine whether it's just a benign polymorphism or a real abnormality is that there are a few databases out there. That one paper that was published in Nature, they have all of their polymorphisms deposited in a database. So you can look to see if it's been described as a copy number polymorphism previously. That's one thing that we do. And then the other thing is to try to get both parents and see if one of the parents also carries that change. So that right now, that's the way we're dealing with that. Harvey?
Have you tried using parental DNA as the
Reference DNA? We have not tried to do that. I mean, it's just a little bit impractical, for one thing, to draw parental bloods, to do the testing.
You're getting it anyway, or you only get it when there's an abnormality?
Yeah, we're not going to ask for it up front. I think there are a few labs that are doing prenatal testing because they're doing it home brew that may ask for parental DNAs up front. But we are not, at this point, asking for that. Yeah, Dan?
So for Karen, the gene expression array business, there are people who do two-color hybridizations, and also if you like use Affymetrix chips, it's a one-color thing. And I guess one advantage of the one color is that you don't have to have a reference sample which could potentially introduce its own error and artifacts. Is there any technology that people are working with to do one-color Array Cgh?
Affymetrix has done one-color Array Cgh.
Does it work?
It's noisy, and for Array Cgh, the two-color hybridizations work very well. So
DNA reliable enough that you can use it all the time?
well, we buy commercially available pooled reference DNA from Promega, so yeah.
What is the potential of using the array prenatally?
Well, like I said, right now it's limited to just a few labs that have their own homebrew assays, because anyone using, most of the other labs that are using the spectral genomics techniques, we had agreed not to use it for prenatal testing. So there are only a few labs that are running homebrew assays right now that are doing prenatal testing. I think sometime in the future it will be useful for prenatal testing. I think it's going to take a while. It's going to take some getting used to.
We've run out of time, but if anybody has additional questions for Dr. Tsuchiya, maybe she'll stick around for a few minutes. I just want to thank her again for a great talk.
Thank you.